Today we are going to study about nucleic acid part 5. In that we'll study about the structure, function and types of RNA. So structure of RNA. In the last video only we learned that RNA is a single stranded molecule, but wherever the palindromic sequences, inverted repeats, polypurine or polypyrimidine tracts are found, they pairs among each other and form a helix structure. The helix conformation the RNA take is the A form of helix. Now as you can see in the figure, this is a single strand, but because of having the complementary sequences, inverted repeats and all, it pairs in the same strand. So like this particular stretch, if you see, they are complementary and hence they form double helix here. This A, because no complementary is present, therefore it bulges out. This is the internal loops. Why this loop is formed? Because this section is not complementary with each other, so they cannot form the bond. Similarly, the hairpin formation, as we studied, because of palindromic sequence or inverted repeats, they form the hairpin structure. So the structure of RNA, it takes the A form of helix, and wherever such sequences are found, like palindromic sequence, inverted repeats, stretch of adenine, polypurine or polypyrimidine tract, it forms bulge, loop, hairpin. Okay. So this is the structure of RNA. Now sometimes what happens? They form the pseudoknot. Pseudoknot, as the diagram is showing, here some base is there, here some base is found. They are pairing with this bases like this. So such type of knot is known as pseudoknot. So base pairing when happens between sequences that are not contiguous to form complex structure are known as pseudoknots. Now this is the structure of tRNA. As you can see, the tRNA, the complete length is 72 to 76 nucleotide long. The tRNA is. It has following arms, that is acceptor arm, T arm, variable arm, anticodon arm and D arm. tRNA is a transfer RNA. What it transfer? Amino acid according to the codons found on mRNA. So the amino acid binds with the acceptor arm of tRNA. Anticodon arm, it pairs with codon of mRNA. Then only it comes to know that which amino acid to bring. T arm, because this arm contains an unusual base which is known as pseudouridine. That's why this arm is known as T arm. D arm, this arm is called D arm because this arm also contains one unusual base, that is dihydrouridine. So tRNA contains unusual bases, and because of this only the arms are labeled. This is the 2D structure of tRNA. If we have to make a 3D structure of tRNA, it takes some L-shaped, L form. So like this, it will form some L-shaped structure in 3D form. And this 2D structure is known as clover leaf model. So the main RNAs we studied till now is mRNA. The function is messenger RNA. It carries the message from DNA to make the protein. tRNA, transfer RNA, it helps in translation process by transferring amino acids. rRNA, it forms the ribosome, which is known as the protein factory of the cell. So all the three RNAs help in translation or in protein formation. Apart from these RNAs, the other important RNAs are small nuclear RNA, which is 150 nucleotide long. This helps in splicing, RNA splicing. What is splicing? We'll study in depth in molecular biology, but for now splicing is from DNA. When mRNA is made, the first transcript which is formed is known as primary transcript. It contains many useful and unuseful messages. So the part which are not useful are removed to have only the part which can form protein. So the mature RNA is formed after removing the parts which are not needed to form the protein. So the mature mRNA is always shorter than the primary transcript. So this process is known as splicing, so that the useful informations can be joined together and the part which is not responsible for forming the protein can be removed. So this is known as splicing. Next is small nucleolar RNA. It helps in the modification of genetic material. PV interacting RNAs. They help to maintain the stability of genome, especially in germ cells. MicroRNA. They help in gene regulation, gene silencing. Gene silencing means to stop the gene from expressing so that the protein should not form. So when any gene is stopped from getting translated is known as gene silencing. Okay. So this is one mechanism by which the genes are regulated. In many diseases also, like if the expression of any gene is causing that disease, then we can silence that gene to cure that disease. Next is long non-coding RNAs. These long non-coding RNAs are also help for gene regulation. So these are the other important RNAs and their function. Apart from this, there are multiple other RNAs found in a cell. Clear? So now at the end, what all we can say, we studied the function of RNA. That means RNA is having the regulatory function. RNA forming the ribosome, that means structural function. RNA can act as an enzyme, ribozyme we call it as, correct. So RNA has multiple functions. Coming to DNA, the function of DNA is to form protein. Whatever protein in our body is formed is because of the DNA. Next, transfer of genetic information. It helps to transfer the genetic information. Apart from this, if we separately talk about the nucleotides, then the nucleotide function is definitely nucleic acid as cofactors, as intermediates of signal transduction pathway. So these are the functions of RNA, DNA, or separately as nucleotide. Now the question is, you have to find the name and the function of other types of RNA which I have not mentioned in this video. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.